Aloha everybody. Last time I ended my presentation by introducing the four complexes involved in the electron transfer pathway of oxidative phosphorylation. Today I will go over the detailed reactions of each of these complexes and how they're connected together, and from there what is the difference between electrons carried by NADH and FADH2, and also how the electrons get channeled to these to the electron transfer pathway, and ultimately that will determine how much ATP will be generated per electron pair. Alright, so let's start with complex 1, NADH dehydrogenase. This is a very large complex. If I'm not mistaken, this is the largest of the four. The two main reactions that this complex catalyzes is the transfer of electron pair from NADH to Q, converting ubiquinone to ubiquinol, or converting Q to QH2. And the second reaction is the transport of 4 protons from the mitochondrial matrix to the mitochondrial intermembrane space. This is the space between the outer and inner membrane of the mitochondria. Alright, now how are these electrons actually transferred, or how do they get from NADH to Q? So from NADH the electrons are passed on to FMN, and from FMN to a series of iron sulfur proteins, to N2 iron sulfur protein, and then from there to Q, to ubiquinone. And in this process, though it is not exactly clear how this happens, 4 protons will be transported across the inner mitochondrial membrane. The four protons will be transported simultaneously. But again, like I said, it is not exactly clear how that happens, because where these protons are transported, the location of their actual the transport sites, and where ubiquinone is converted to ubiquinol are quite far away. If I can say that, since we're talking about such small distances, but nonetheless, the sites are far away from each other, and even though it's believed that somehow Q is involved in this transport, it's not exactly clear how that happens. And one other thing, in addition to the transport, 2 protons are actually picked up from the matrix to form ubiquinol. All right, complex 2, succinate dehydrogenase. The name should be familiar to you, because this enzyme has been previously discussed. And if you think carefully, you would realize that this was the same enzyme that is part of the TCA cycle. This is the enzyme that converts succinate to fumarate, and an electron pair, the only electron pair is from the TCA cycle, is transferred to FAD, converting it to FADH2. And we will see now how the fate of these electrons is actually different compared to the electron pairs loaded on NAD+. Alright, complex 2 has 4 subunits. The C and D subunits are transmembrane subunits. They contain the ubiquinone site. The A and B subunits are the matrix subunits. They form the binding site for succinate and for FAD. Now again, the two reactions that are catalyzed or that are furnished by this complex is the transfer of electron pair from succinate, which is the substrate in this case, to FAD, and then from there to iron sulfur proteins, and then from there to ubiquinone, converting it to ubiquinol. Something that is noteworthy to be mentioned in this point is that no protons are being transported across the inner mitochondrial membrane. All right. Knowing that, is there something in this process which could explain why electron transport from the succinate reaction in the TCA cycle ultimately gives less ATP? I mean, I already mentioned it, that electrons loaded on FADH2, if you notice correctly, they are loaded on Q by complex 2. They basically skip complex 1. And complex 2 does not transport protons to the matrix. I mean, from the matrix to the intermembrane space. And I've mentioned this before, but ultimately the proton gradient generated across the inner mitochondrial membrane will later on be used by ATP synthase to generate ATP. So the fact that the electrons that are loaded on FADH2 skip complex 1, which transports protons and contribute to the generation of the gradient, and these electrons are loaded instead they're loaded on Q instead through complex 1, but through complex 2, skipping any protons transport, eventually would lead to a smaller gradient, or it will not contribute as much to the gradient as those loaded to complex 1, and the result of ATP will be much, the resulting ATP synthesized will be smaller, or less, I should say, per electron pair. All right, this is going to make a little bit more sense now, a little later when we see the whole picture. Now, for now, just know that electrons loaded from FADH2 from succinate dehydrogenase skip complex 1. They are loaded directly on Q through succinate dehydrogenase, and succinate dehydrogenase does not transport protons across the inner mitochondrial membrane. All right, so complex 2 channels the electrons from the TCA cycle, these electrons that have been loaded on FADH2 directly to Q. Again, see, it's skipping complex 1. Well, what about electrons that are loaded on FADH2 from fat degradation? Well, if you remember, lipids, the triglycerides, have a glycerol portion and fatty acid portion. And if you look back in the things that we've learned, the glycerol is processed first by phosphorylation, and then it's the glycerol triphosphate is converted to dihydroxyacetone phosphate, which enters glycolysis or gluconeogenesis for that matter, based on what's needed. And the enzyme that catalyzes that reaction was glycerol triphosphate dehydrogenase. That same enzyme can donate electrons through its FAD cofactor directly to Q. The first reaction of beta oxidation, the acyl-CoA dehydrogenase, it also loads electrons on FAD, converting it to FADH2. Now, how will these electrons enter the electron transfer pathway? Well, they are transported from the acyl-CoA dehydrogenase to another electron transferring flavin protein, and then to another electron transfer flavoring protein that will transfer it to iron sulfur centers, and they will transfer later on to Q, converting it to QH2. So, what is common for the substrate to FAD electron transport? FAD from TCA through complex 2, FAD from beta oxidation directly to Q, even circumventing 1 in complex 2, and electrons from glycerol degradation or FAD molecules that are in the cytosol, they come in to glycerol triphosphate dehydrogenase, again loaded directly on Q. So what is common so far between all of them? none of these processes leads to a transport of protons from the matrix to the intermembrane space. In other words, so far these electrons have not contributed for the establishment of a proton gradient that can be used later on for the synthesis of ATP. That is something very important for you to realize and remember. All right, complex 3. Structurally, complex 3 is made of two identical complexes. Each of these complexes is made of 11 identical, well, not identical, 11 subunits, but the two complexes made of 11 subunits are identical. Let me clarify that. You have one complex made of 11 subunits, and then another complex made also of the same 11 subunits, and then they bind together, and that's how complex 3 is formed. The purpose of complex 3 is to transfer electrons from ubiquinol, or QH2, to cytochrome C. This is known as the Q cycle. So let's see what the Q cycle does. And during this Q cycle, 4 protons will be transported from the matrix to the inner membrane space. All right. The QP site right here, it's P for positive, is the site where ubiquinol will first bind. Then once bound, it will donate, and this is the interesting part now, it will donate 1 electron to a risky iron protein, which will then donate that electron to cytochrome C1, and then from there to cytochrome C. The other electron from ubiquinol will be donated to heme Bl, then heme Bh, and then from there to a molecule of ubiquinone, converting it to semiquinone. The 2 protons carried by ubiquinol are released in the intermembrane space. Now, when that is completed, another molecule of QH2 will come in. It will bind the same site, and it will repeat exactly the same thing the previous did. It will donate 1 electrons to the risky iron protein, to cytochrome C1, to cytochrome C. And then the other electron to heme Bl, heme Bh, and now to the semiquinone that will pick up 2 protons from the matrix and become ubiquinol. And that ubiquinol will be released. So the net product of this reaction is, well, before I get to the net product, what happened in this reaction, we used 2 molecules of ubiquinol. We generated 2 molecules of cytochrome C, each of which carries 1 electron. 4 protons are released in the intermembrane space, and 1 molecule of ubiquinol was regenerated or produced. So the net product is 1 Q, 1 ubiquinone, 1 ubiquinol, 4 protons across the inner mitochondrial membrane, and 2 cytochrome C, each one of them carrying 1 electron. That is the Q cycle. And the reason you might wonder, well, where's the cycle? You can think of this product as the one that keeps cycling. That second ubiquinol that is needed is basically represents the cycle. Because this regeneration will swing back and come back again and the second time, and just keeps the Q cycle going. All right. The next reaction is now the offloading of the electrons from cytochrome C to complex 4, or cytochrome oxidase. Again, large complex. The 2 reactions that catalyzes is the transfer of this electron from cytochrome C to molecular oxygen to form water, and in the process, protons are transported across the inner mitochondrial membrane. Now, how are the electrons transferred from cytochrome C to a protein that contains copper ions, and then from there to iron copper centers, and then eventually it would reach the oxygen that gets reduced to water. Now, I want to focus your attention a little bit on this representation. It shows here that 4 protons are being transported. And if you're not careful, you would get the idea that, yeah, complex 4 transports 4 protons per electron pair. But that is not the case. Notice how many electrons are being represented here. 4 electrons, that's 2 electron pairs. For 2 electron pairs, 4 protons are transported. In other words, 1 proton per electron transported by cytochrome C. In other words, 2 protons per electron pair. That would only make 1 molecule of water. But again, that normally doesn't happen because you don't split a molecular oxygen to 2 atoms. These are very reactive compounds. So you shouldn't be thinking it in this way. Mathematically, on a piece of paper, yes, you can represent it as half a molecule of oxygen, but that in reality never happens. This molecular oxygen will be converted to water, to 2 molecules of water, which will require 4 electrons donated from cytochrome C, and which will transport 4 protons. In other words, this image represents what happens to convert, what's needed to happen to convert 1 molecule of oxygen to 2 molecules of water. But per electron pair transported, 2 protons will be transported into intermembrane space. All right. Now, 2 electrons are transferred from NADH to oxygen. Complex 1 transport 4 protons. These electrons will skip complex 2 because complex 1 loads it directly on Q. Then Q offloads its electrons to cytochrome C, and 4 protons are transported. Cytochrome C, as you know, it's soluble, so it's free, and then it binds complex 4 and offloads its 2 electrons, and as a result, 2 protons are being transferred by complex 4. So the total per electron pair loaded or carried by NADH, the total protons transported from the matrix to the intermembrane space is 10. 4 from 1, 4 from 3, and 2 from complex 4. Again, 4 protons from complex 1, 4 protons from complex 3, and 2 protons from complex 4. Now, what will be the net proton transport for electrons donated by FADH2? Remember, how are these electrons passed on Q? Complex 2 or the other additional enzymes that can offload electrons to Q. They all skip complex 1. Complex 2 doesn't transport any, or any of the other enzymes don't transport any protons across the inner mitochondrial membrane. So the total number of protons transported per electron pair carried by FADH2 is 6. 4 from complex 3, 2 from complex 4. All right, now the oxidative phosphorylation pathway is actually a very lucrative, if I can use that term, target for drug development. I mean, if you think if you somehow stop the energy production of a cell, or especially if it's a bacteria or fungus or anything else that is microscopic, you would be able to control its progression or its life. So that's why there's so much effort. Well, a lot of effort has went to generate blockers of these pathways with the hope that they eventually would serve as medicinal, will have medicinal purposes for treatment of diseases. I mean, some of the most potent toxins act at the electron transport pathway. I mean, cyanide, even carbon monoxide, which is not it's a naturally occurring toxin, so there's a cyanide, but you know how effective these toxins are in terms of how toxic they are, but that is because they inhibit the transfer of electrons from cytochrome C to complex 4, and as a result, their toxicity is really high. And you can see a fair amount also of antibiotics and other compounds that can be used as blockers of different steps of the oxidation phosphorylation pathway. I usually also show you this slide. I have absolutely no intention of you to memorize anything here, but I wanted to also point out that even though we're talking about these normal, wonderful processes that occur in the body, how energy is generated, in this process, we have actually a fairly reactive compounds that are made. A semiquinone, as you know, is a free radical, and it's highly reactive. And if it reacts with oxygen, and it does react with oxygen in the blood, even though oxygen is low concentration, that oxygen basically gets turned into a radical. And these reactive oxygen species are highly damaging for proteins, for DNA, especially proteins, since they're around proteins in the cytosol or in the matrix, actually, because this one shows in the matrix. So even though this process is wonderful and it generates energy, these reaction oxygen bodies do damage proteins and cells. However, the body does have a system to reduce that oxidative stress in the mitochondria and in the cell, and that's how that effect is counteracted. But again, it still has a damaging effect on the cells and on the body. But this is something we cannot avoid. And this is part of the aging process that we all experience. Perhaps that's why so many legitimate and not legitimate companies focus on promoting us taking antioxidants as pills, claiming that these pills will probably counteract these effects. Whether that really happens, I have no idea. And since I'm usually a little bit more on the skeptical side when we talk about these type of stuff, I would imagine that their effect is way exaggerated for, of course, sales purposes. All right, let's move on. Now, so far I've shown you how NADH offloads electrons, but that NADH is in the matrix. But NADH from a cytosol cannot just simply get to complex 1. There's actually no easy way for it to go through the membranes, especially the inner mitochondrial membrane. However, the body has established a pathway that channels NADH from the cytosol. Here it's shown in intermembrane space because NADH passes through the outer mitochondrial membrane. It just cannot pass the inner mitochondrial membrane. And so this mechanism, called malate-aspartate shuttle, will move NADH from the cytosol to the matrix of the mitochondria, and then that NADH can offload its electrons to complex 1. Now, how does this happen? Well, with very familiar enzymes. Oxaloacetate in the cytosol, with the help of malate dehydrogenase. Remember, this enzyme can convert oxaloacetate to malate with the help of NADH, converting it to NAD+. Now the malate will be transported in the matrix in exchange of alpha-ketoglutarate that comes out. Once inside the matrix, same enzyme, malate dehydrogenase, comes in, oxidizes malate to oxaloacetate. NADH is now regenerated, but inside the matrix, the resulting oxaloacetate gets aminated to aspartate aminotransferase, transferring amino group from glutamate to oxaloacetate, forming aspartate and alpha-ketoglutarate. Alpha-ketoglutarate goes in exchange with malate, like I pointed. The resulting aspartate now will be transported by a different transporter that exchanges aspartate for glutamate. Aspartate comes out, glutamate comes in. Aspartate now is converted again to oxaloacetate by aspartate aminotransferase. And then alpha ketoglutarate is aminated, converting it to glutamate that goes in exchange with aspartate. So you see, it's a very well designed cycling process that has a net product of moving NADH from the cytosol to the matrix. So was there something spent in this process? Was there ATP spent in this process? Examine it carefully. Someone might say NADH. Well, no, we spent it, but then we regenerated. So no net change in NADH. It's just a position. Was there anything else spent? Well, I'm sure some binding energy, but nothing really major. There was no ATP used in this process. How many protons would be transported now per electron pair donated by NADH that was transferred from the cytosol to the matrix using this pathway? Where will this NADH offload its electrons? If you think that it was complex 1, you're correct. If you think that it's complex 2, you're wrong. They go to complex 1. And the resulting number of protons transported from this electron pair is 10. There is also another way for NADH that is in the cytosol to offload its electrons to Q. And that is through a familiar enzyme, glycerol 3-phosphate dehydrogenase. Now, where do we produce NADH in the cytosol? In glycolysis. The NADH in the cytosol produced during glycolysis will be oxidized, and the electrons will be transferred to dihydroxyacetone phosphate, converting it to glycerol 3-phosphate. The cytosolic glycerol 3-phosphate dehydrogenase catalyzes this reaction. Now, the glycerol 3-phosphate, with the help of the mitochondrial glycerol 3-phosphate dehydrogenase, now will be oxidized, transferring an electron pair from glycerol 3-phosphate to FAD, forming FADH2, and then dihydroxyacetone phosphate, which goes either for glycolysis or gluconeogenesis. But that electron pair now from FADH2 gets transported directly to Q, forming ubiquinol. And then from there to complex 3. So, was there something spent in this process? No, no ATP was spent. How many protons will be transported per NADH molecule that offloaded its electron pairs following this pathway? From NADH to dihydroxyacetone phosphate to FADH, forming FADH2, and then to Q. Now, think about it. We skipped complex 1. Complex 2, whether we skip it or not, doesn't really matter because it doesn't contribute with proton transport. But we skipped complex 1. This electron pair, even though it's carried by NADH, because it doesn't enter the mitochondria, but transfers its electrons straight from the cytosol to Q, will only be the reason for the transport of 6 protons from the matrix to the intermembrane space. Now let's get back to glycolysis. How many ATP molecules are produced during glycolysis? If you remember, 2 ATPs were spent in the preparatory phase, 4 ATPs were produced in the payoff phase, and 2 molecules of NADH produced, which is equal to 3 ATPs produced if the electron pair is transferred to the mitochondria by the glycerol 3-phosphate shuttle. This, if the electron pair loaded in NADH gets transferred to Q by glycerol 3-phosphate dehydrogenase, which happens in the muscle and the brain, we will only get 6 protons or 3, I'm sorry, not 2, 1.5 each, 3 molecules of ATP. Now, if this NADH gets transported inside the mitochondrial matrix, into the matrix by the malate aspartate shuttle, then that pair would lead to 10 protons transported across the membrane, and as a result, 5 molecules of ATP. I sometimes say that since we have 2 molecules of NADH, if one goes through the glycerol triphosphate dehydrogenase, we'll get 1.5, and if one pair goes through the aspartate shuttle, malate aspartate shuttle, then we'll have 2.5 molecules of ATP, and the grand total is 4. Unfortunately, I don't think that's how it happens, because the glycerol triphosphate dehydrogenase happens in the muscle and the brain, and the malate aspartate shuttle happens in the liver, kidney, and in the heart. So that's why when we talked about glycolysis, we said that either 30 or 32, not 30, 31, or 32, because it depends on what tissue this happens. And if we look at specifically the tissue or a mixture of different tissues. So anyway, if we talk about a single cell, either we go with 30 or 32 based on the chosen method of transferring electrons from the NADH generated during glycolysis. And that's why right here it says 3 or 5. All right. With that, I will end my presentation today. I hope this was clear. Again, if there are any questions, please let me know. I will be happy to help. Next time, we will talk about one of my favorite topics, and one of the most fascinating topics, as a matter of fact, at least to me, and that is ATP synthesis and ATP synthase. So until next time, though, I hope everything goes well, and let me know if you have any questions.